Okay, hi there. This is John Bishop, and I'm here with my good friend, Dr. Nancy Flournoy. And today we are going to be talking about one of the Lifewave amazing patches called the Eon Patch. And Nancy has a really incredible career. I'd just like a few minutes, Nancy, just to give a little history of your background.
Okay, thank you, John. I'm delighted to be with you again. And I just say I got my doctorate at the University of Washington and in biomathematics, and then had quite a career in clinical research. My expertise is experimental design, and I have worked both in clinical research practice and in academic research. I'm now Professor Emeritus, a distinguished Professor Emeritus at the University of Missouri. That's my appointment, but Emeritus means I don't get paid anymore for the work I do. And so I live in Bellingham, Washington, where I am doing lots of things, including studying these wonderful Lifewave patches. And so with that introduction, I will give a talk about the Eon patch in particular, and then we'll have questions and answers and chat about anything that anyone wants to chat about. So let me share my screen.
Yeah, you're all ready.
Okay, tell me if you can see it.
Yes, we can see that.
Good. Okay. So the topic today is the Eon Patch, and this is one of a series of talks that I have been giving. And I will just point out to start that I have a website, lightpatchscience.com. It's fairly crude, but I have really enjoyed trying to investigate what these patches do. And the website, sometimes it lets me get things off my University of Missouri web files, and sometimes it's a little sticky, but it's my attempt to collect the information as I learn it and put it together in a coherent pattern. So you can, in fact, buy patches off that website, but it's really, it really serves a purpose for me and for, I hope, other people in terms of trying to learn how these patches work and what we know about them. This is the sleeve that the patches come in. You get essentially 30 patches, which if you wore them every day is a month's supply. Some people wear the Eon patch every day. Some people, there are 3 so-called Y-age patches, and some people rotate through them using a different one each day, or some people pick the one that they particularly think they need on a particular day. The claim that Lifewave makes about the patches is here on the front. They can only put, they do not do, it's not a drug, it's not approved by the FDA for treating a disease. So what is on the label is what they are permitted legally to say about it based on the research that they've done. And what they say is it's going to reduce stress in the body, promote relaxation. They point out that it's a patented proprietary form of phototherapy, and that it will transmit no drugs, chemicals, or stimulants into your body. So we're going to delve a little further into what the patches are designed to do and the exact research that has been done to support these claims here. I'll just point out to begin with that the patch was released in 2011, and I put that date in your brain because as we look at the science that surrounds what this patch was designed to do, we'll find out that there is a lot of current activity that's related to it. And it makes it kind of interesting to see that it was designed really ahead of its time, not to mention the fact that the patches are ahead of their time. So I'll just have a couple overview slides for people that haven't heard talks that I have given before, or maybe new to the patches. The Lifewave company has a whole batch of patches. And if I have an overview talk, which you can find on my YouTube channel. If you look for Nancy Flournoy Lifewave patches, you should be able to find it. And it's also on my website, but last time I looked, the overview talk comes up. Some of them are a little sticky sometimes. I have to figure out how to fix that. But the overview talk covers how light makes things happen in the body, a couple of different models of aging. It talks about crystals and electrical conductivity of your connective tissue. Talks about biocommunication systems, the nervous system, connective tissue, the meridians, and then a brief introduction to each of the various patches, which we're now talking about in more detail as in this particular talk. So also as an overview and into the kind of the philosophy, the idea, so this is all electrical. I mean, we've got this patch that's going to take light from your body and reflect it back, infrared light and some in the visual spectrum, but mostly infrared light, and reflect back specific frequencies. And so, why do they think that's going to do any good? Well, the idea is that we are, one view of our body is an electrical view, and there's a lot of electrical activity in everything that makes us function. And so the idea is that as the light produces oscillations, there is going to be a response in the body, and it's going to be a response by molecules and atoms in the body that are of a similar frequency to that that's being produced. And the idea is that that disease is created by an individual getting stuck and not being able to respond. And that when light frequency comes into the body, if it is corresponding to a frequency that's really weak or stuck, it's going to give it a little disruption, and that's going to help it move into a healthy state. So that's the philosophical point of view. Now, the particular aim of the Eon Patch, David Smith told us in a talk last April, a year ago. And he said that it was designed to increase the queen bee peptide in the liver. Well, so that's part of this exploration. We're going to find out what the queen bee peptide is, what we know about it. So that's coming up. And let's see, I've already told you that the patches are designed to influence specific photoreceptors in your body, and that they're going to reflect the frequencies, and that the aim is to increase the products, specific products of metabolism. And this sort of philosophy and this and a longer exposition of it can be found in a paper by Thomas and Helen Brudzinski. Thomas was a professor at the University of Washington in engineering, and Helen is a PhD in psychology. And Thomas has passed away and Helen has retired. So we're very fortunate they did the work that they did. So the target of the Eon Patch is the queen bee peptide. And with a little research, one can find that that has been given the very appropriate name, the chemical name of royal lactin. So here's a picture of it, this curly thing, long, wiggly. It's a fatty acid. And its abbreviation is over here, MRJP1. Well, that stands for major royal jelly protein number 1. And you can guess by the fact that there's a protein number 1 that there's also a 2, 3, 4, and 5. But in 2011, Masaki Kamakura wrote a paper saying that this particular amino acid is the one that is responsible for making the queen bee and responsible for the development of her long life. Now, notice this paper appears the year that the Eon Patch was actually released. So, maybe David had some connection with the research that led up to this paper. But in 2016, a group of researchers contested this. They tried to replicate that study and failed. And they said that their conclusion was that it was not just this peptide alone, but that you also needed numbers 2, 3, and 5 to get the queen to be the queen bee. Now, the story is not over because Kamakura comes back and argues that Buttstead's experiment is faulty and that they didn't consider the right features, and so this is in 2016. He's standing by his statement that one protein peptide is the one that's responsible. Well, there's the story is not. I would say it's not settled. You can find a lot of research between 2016 up to the beginning where they are trying to figure out exactly what it is that makes the queen and what's necessary and what's not necessary. And so research on these queen bee is really kind of a hot area, which is interesting in and of itself. So over here on the right, you can see that a peptide is a chain of amino acids. And I've got this little structure here. It's kind of a generic structure. And you have the amino acids are long chains with lots of carbons in them and a kind of, shorthand here. Let me see if I can back up. And then this particular queen bee peptide, the peptide has 2 groups hanging off. Well, really 3 groups hanging off the end, and these groups differ depending on what kind of peptide it is. So that's your little chemistry for the day. The queen in particular was going to grow more cells than the average bee, and the other feature that she has is she can regenerate, whereas the other bees cannot. And of course, for anti-aging, we're interested in regeneration. So, it wasn't until 2018 that a group from Stanford actually found that the royal jelly proteins, that's what we're talking about, of which the queen bee one is one. These were found in humans in 2018. So that's really just a few years ago, really pretty stunning. And what this group from Stanford and other research places discovered is that the proteins promote division of stem cells, which are the cells in our body that can go on to make anything. And so, there's a lot of people that pay a lot of money for still cell injections, and it is determined that royal jelly is going to help those divide and grow. It also will boost complete regeneration in damaged and absent tissue. And in particular, it's going to complete regeneration in tissue that are where the stem cells are depleted, and that's what occurs with aging. Now, this protein's called royal lactin in the bee, and they named it Regina in mammals. And that was what Derek Wan and his colleagues did in 2018. But if you look at the research going forward, it seems like this royal lactin name has stuck, whether it's in mammals or not. And I've not seen a lot of mention about the mammal analog being called Regina, but you might find it in under either name. So they did establish in mice that the royal lactin Regina peptide induces this stem cell-like state. So that was the mammal they worked with, not human beings. Oops, I think I went too far. There we go. Okay, so what do we know about the royal jellies, which is this combination of peptides? So on the left, I have sort of the highlights from a review paper by Ramanathan and his colleagues in 2018. And he points out that the royal jelly is extremely nutritious and that people have been eating it since prehistoric times, but that in addition, it's now understood to influence gene expression, and that it's been shown to have effects in animal models as well as the bees. The third bullet point is that these proteins are responsible for the development, Cas differentiation. That's a little more general than just making the queen, longevity and reproduction in bees. And that it also has antibiotic and bioactive compounds that make it a great nutriceutical if you want to go out and buy bee honey, I mean, special kinds of bee honey. So here's another paper that talks about its major anti-aging activity, and those points are summarized in that it's been shown to lower blood pressure, lower fats in the blood, stop tumor growth, it's anti-inflammatory, and it modulates immune response. So now, this may seem like a little digression, but we will come back, it'll pull together in a minute. I want to talk about two amino acids. One is glutamate, and one is known as GABA because it has a very long name. Everybody calls it GABA. And you can think of these as the gas and the brake. So glutamate is the primary thing that excites you. It gets you going. It stimulates your nerve cells. It makes you ready to receive information. It regulates the generation of new neurons, and it plays a role in learning and memory. GABA, on the other side, is the primary inhibitory neurotransmitter, and it blocks signals sent between the nerve cells and the brain or the spinal cord. So one gets you revved up and the other slows you down. And you can see in this little picture at the bottom that you want your glutamate and your GABA to be pretty well balanced so that you are going along at a nice pace. And if you have too much, if your GABA pulls you down, then you're going to be anxious, you're restless, you can have migraines, tics, different kinds of weird things can happen to you. So that's sort of simple, but it's actually just a little more complicated. It's not a matter of just having them equal. You have to take time into account. So here I have a little chart that goes from, early in the morning to late at night. And the green here is ideally what a glutamate should do. That is, it should go up and come down so that it peaks around 2 o'clock in the afternoon. And glutamate's really good to be accelerating when you're getting up to speed because it helps you go in the morning. And you need it to come down so that you can go to sleep. And on the other hand, you have GABA, which is high in the evening when you really need to be sleeping, and you want it to come down in the middle of the day so you can be out there getting things done. And you can see how things can go wrong. You can see these two have to go together. That is, if glutamate doesn't go high enough, you never get to feel like you're going, even if you have all the ATP you could get or need. And there's all kinds of permutations of how these two curves could go astray. One of them gets stuck high, one of them gets stuck low. Either way, it's a bad deal. And in particular, if you take supplements, it's like hitting the brake and the accelerator at the same time if you take the GABA supplement. So that's not proved to be a very useful way to try and regulate your wake-sleep cycle. And I just noticed that PTSD is like having too much glutamate during the day and not enough GABA to bring it down. So it just stays high, and the person with PTSD is always revved up and ready to go. This little figure up here is autophagy. That is one of the reasons you need to sleep. You need to regenerate and repair when you're sleeping, but you also need these sort of cannibal cells that go around and eat up all the things that are all the dead cells and things that need to be taken out of your body and move them away. And all of that has to happen at night. So here's a trick. I mean, GABA and glutamate don't operate independently. If you start over here with this amino acid that's called glutamine, glutamine makes glutamate, and glutamate makes GABA, and then they go on. So, and glutamate can go back and make more glutamine. It can go in a circle. So these two molecules are very intimately, the availability of them and the number of them are very intimately wound up with each other. That was just the point of that slide. So now what's that got to do with bees? Well, and with the queen bee peptide in particular. So it just happened that the week I was preparing this talk, we have a friend who's a nutritional biochemist. And here I am trying to figure out what in the world the queen bee peptide is and what it does. And this friend who's the nutritional biochemist calls me up and starts talking about the honeybees and the jelly that they make and why it's so incredible. And so his story is the one that gives you the bee and glutamate GABA connection. And if you look down at the bottom of the screen, we are going to come to a Lifewave study that actually supports this theory that I'm telling you. So the way he motivates the connection is that honeybees are really miraculous in the way they handle being awake and being asleep. That is the switch between the two, the going back and forth between the two. That is, your honeybee goes out to get the pollen, essentially runs a marathon, comes back, sleeps for 15 minutes, and then goes again. So one of the things, the thing in his mind that ties the honeybee to this switch between the GABA and the glutamate is that they really have to maintain the sleep-wake cycle. They have to have really good control on how these two amino acids are playing together. And he notes that the mammals that do a similar, that have similar control over their wake sleep switch, being able to turn it on and off very quickly, are the whales and the dolphins, because if they can't wake up quickly, they risk drowning because they don't breathe underwater. So given this connection, we turn to the second part of this talk, which is to look at what the Eon patches have actually been shown to do. So what we have up until now is this statement by David Smith that he designed the patches to stimulate the production of the queen bee peptide. And we've learned a bit about, if in fact the production of the queen bee peptide, or peptides plural, was stimulated, what that might be doing based on the biochemistry and the biophysics that's known. And now we're going to look at what they've actually tested in the patch. And of course, what they can actually test on the patch is limited to what they can actually measure. And so the mapping of the goal and the evidence is not always exactly what we would like it most to be. But what I was mentioning before, which is particularly exciting about this little theory that my nutritional biochemistry friend postulated, is that the Eon patches have been shown to increase amino acids. And in particular, if you look here at this table of amino acids that have been shown to be enhanced, the top two, the glutamate and glutamine, well, glutamate makes glutamine and glutamine makes GABA. So that whole day-night cycle is supported by this evidence that the fact that Lifewave patches would actually help control that switching mechanism and that balance is a rather simple step away from knowing that it does enhance the production of these amino acids separately. I mean, there's other things that could mucky up the works, but this is kind of exciting. So this is a work by Melinda Connor, Dr. Connor, and her colleague Eckhoff in 2013. And they studied a lot of things. It almost looked like they were studying too many things, that they were going to get some things to be significant just by chance. But what was impressive of all this catalog of things that they studied, it was the batch of neurotransmitters that all, that came out significant. And of all the other classes of things that they looked at, the other things didn't seem to be affected at all. So that was pretty cool. And okay, so the amino acids affect neurotransmission. That's the context in which we've been talking. They also help with protein and collagen building, with detoxification and immune function. So we've talked about the first two. I'm going to summarize the others just very quickly because of time limitations. And if you want to come back and look at these slides, you're welcome. But L-DOPA, it makes dopamine, which is kind of a happy drug in your brain, and one could talk a lot about that. It mediates the release of neurotropins, and those are molecules which promote the survival of neurons, particularly in your brain and your nervous system. And they prevent the neuron from starting what's called programmed cell death, which is we could have several, we could talk for a while about that, but it's I'll call it a theory, but it's pretty well established that if you have a group of cells and they multiply, that after they've multiplied a certain number of times, they basically go quiet and they say, I'm finished now, I'm going to quit. So this stops that, and it induces the cells that are going to form more neurons. So the other amino acid is called serine, and it's important for brain development and for the synthesis of not only proteins, neurotransmitters, nucleotides, and lipids. So that's almost everything. It's a source of what's called units that are needed for methylation reactions. And there's a whole class of things reported to health that are important for health that are related to this process called methylation. It is a mechanism that can control gene expression, and it can change DNA activity without changing the sequence. So it boosts immune function, promotes sleep, fights chronic fatigue, and may be neuroprotective. The last two amino acids, cysteine and proline. Cysteine is a building block for glutathione, which is the master antioxidant in our body. And one of the YH patches by Lifewave is glutathione, the patch to stimulate the production of glutathione directly. Cysteine is used to construct proteins. In particular, it'll bind with metals such as mercury, lead, and cadmium. So it's good for detoxing and also for alcohol toxicity, because it turns acetyl, I can't say it, into, anyway, it's good for alcohol toxicity. And proline's good for collagen. So it's good for your skin, your tendons, your bones, your connective tissue, and also your joints. Okay, well, that was just a mouthful. So we have a few other studies, which I will try to express succinctly. But one is to go back to what the Lifewave company advertised on their little sleeve, which is all about relaxation. So where do they get this idea that, where do they get the evidence that that the patch causes relaxation, which is what they then put on the cover of their product? Well, stress, as is in the autonomic nervous system, is one thing that can be measured. So they measure it. So on the right-hand side, we actually have the components of the autonomic nervous system here. It's got 3 components. I didn't see them study the last one at all. So they did study the parasympathetic system, which is responsible for your resting, your digesting, all the things you do when your body is at rest, and the sympathetic nervous system, which is responsible for the stimulating side of your activities. And it is in the parasympathetic system that they found the effects of Lifewave in more than one study, but here on the left, we have one in particular, which was done by Dean Clark in 2010. And he patched subjects for 12 hours for 5 days and took infrared measurements using thermal imaging. And at any rate, he concluded that so the thermal imaging, I looked for a picture there. Dean Clark did this kind of study for a number of patches. And for some of the papers, there are pictures that are pretty dramatic of how the subject can be, literally too hot and inflamed, and they come out like all red, and then they get patched and they go to look much more blue, which is cooled down. But anyway, I didn't find any pictures for this particular patch, but his conclusion is that there was a rebalance and that most of the participants, it cooled when they were too warm and warmed when they were too cool. So I just have to take his word for that. Another thing that they can measure is called heart rate variability. And if you look at the little graph on the left, this is a graph of the middle 50% of heart rate variability, and the x-axis is age, years. And first I have to say that a lot of heart rate variability is good. That confused me at first, because when you think of all the little heart charts in the hospital and everything, you think you want a nice steady pulse, you think you want everything to be nice and smooth and regulated. And you go back to that beginning slide that I told you about, is that variability is good, and being able to have respond quickly depends on things being variable. And that's true with this heart rate variability measure, which is actually a ratio of high frequencies to low frequencies. And then it's a complicated electrical formula involving Fourier transforms and such. So it's not, and so it's not easy to say exactly what the y-axis is, but you can certainly see that at 20, it goes from 60 to about 105. And by the time you get to be 55, it's going from, 30 to 50. So it is certainly your flexibility as measured by the heart rate variability value is really reduced by most of us as we reach middle age. So they did 3 studies, and the first one, 20 subjects, they looked only 15 minutes after patching, and they saw an increase in parasympathetic activity. And then they, in 2013, Melinda Connor and Eckhoff looked at only 10 subjects. Now here they're doing case control, and they looked from day 1, which was the baseline, to day 2, and they found no significant difference. So now we have kind of conflicting results. And so there's a third study with 50 subjects that are randomized in a double-blind placebo-controlled study to try and clarify the issue. And they were observed after 20 minutes, looking at the differences between in each group. You look at the change from after a beam patch to your baseline, and then the differences between groups. And they concluded that the parasympathetic response was indeed enhanced. So it still seems like a very short period of time, but I know people that just rave about the Eon Patch and do notice its effects very quickly. My personal experience is that I believe it's effective, but I did not have that dramatic result that said it was my go-to patch. And I do know people for whom it is their go-to patch. So there's another piece of equipment that they have access to, which is this, looks like an MRI scanner where you sort of get in and get hooked up with electrodes, and it's called Electro Interstitial scan. And so it's actually looking at conductivity like an EKG, but it's looking at conductivity of the fluid, the interstitial fluid, which is, all your cells are in fluid, everything's in fluid. So you want your electrical charges to be able to get through the fluid. And the voltage is going to drop when the current meets resistance in the tissues and the fluids. And if you have studied what's normal, you know what kind of measurements you expect to be going through what tissues and what fluids. And they did study a large number of, quote, normal people. And then you can see whether things are out of the normal range. So this study was done by Blake Greenberg and Nazarene. Nazarene's a professor of engineering. Well, he's retired now, but he's a professor of engineering at University of Texas, I think at El Paso. And he has done quite a bit of work, research on these patches. So they patched 12 hours a day for 2 weeks. And they looked at the end, they had two groups. One was patched 3 days a week, the other was patched 5 days a week. And they have a batch of organs over here that they evaluated. And so the ones that were patched 3 days a week showed significant differences in the adrenal glands and the hypothalamus at the end of 2 weeks. And the ones that were patched 5 days a week showed differences in basically everything that they evaluated. So notice there that they didn't patch anybody 7 days a week. The patches, that's light. And so if you get too much of a patch, your body will become attenuated to it and it won't work on you anymore. So that's the reason it's 12 hours a day and not 24 hours a day. And that's the reason that the maximum they looked at was 5 days and not 7 days a week. It's to give your body time to forget about that light signal. So when it gets it again, you're going to react. Now, another study was done by Streeter, and they found changes in just some of the organs, but well, I guess I can't tell you why. It's just different. And then the final study down here by Patog, they did not find changes, but they looked after just 12 hours. So it was a much shorter time frame. There's really so many variations on all the scheduling that could be done that I would really like to see Lifewave. I mean, I know they can't do everything because it's very expensive, but I would like to see some optional studies performed with distributors being volunteers. I would think people would like to do that to get more information about scheduling. So I think we're getting close to the final slide. Another piece of equipment, you notice all this equipment specialized to be able to measure the things, frequencies and things at the level that you need to measure them for the kinds of responses that they're looking for. So this gas discharge visualization, it's a French machine. It's, I don't know the right words, right? At any rate, it's French. It's used in a lot of countries and it's used here, but it's not officially approved here, I think is the right way to say it. But here's a picture. You have a plate. You see, here's glass, and then you have a piece of metal, and then you have the insulator and the metal. They use gold actually because it transmits frequencies better than other things. And then at the top here, you put a leaf on it or something that you want to measure. And if you can see this picture down in the corner, here's what the leaf looks like. This is a nice healthy leaf. It's got a real bright glow. And when they say gas discharge, for the most part, they're talking about water. This is just your natural breathing of your tissue where you evaporate a little around your body. And if you're nice and healthy, it's going to show up as a nice, healthy part of your respiration. And if you're not, it's going to look pretty sick. And there's a little stylized picture down here at the bottom where I'm going the wrong way. Where you see, sorry, I got the bar where I have all your pictures is right in the way of where I'm trying to look. So I'm having a hard time seeing what I want to show you. But if you look in the lower right-hand corner, you see you pretend like on this glass plate, you've put your hand down on it. So now it's actually taking those pictures of your fingertips. And so if you have a fingertip where the light is coming out all around, like the picture on the right, that's a nice healthy picture. And if it's only coming out in some parts of your finger, or it's coming out crooked and erratic, then that's not so good, and it indicates some blockages. So I will just skip through this quickly. Two studies, they both found differences. Streeter concluded that detoxification process was engaging within 24 hours. And I'll not try to say more about that. This is just to say a lot of the studies included, questions to the people. How are you feeling now? How are you feeling later? Did you like it now? Did you like it later? Et cetera, et cetera. And so there was a lot of self-reporting data, which I don't like as much as as the numerical data, but they did conclude that based on it, the patches had a great effect, and it's up to, as I say, I like the numbers and the measurements better. So I have some references about things and the disclaimer that Lifewave is not designed to treat diseases. It's designed for your health and your well-being, and that if you have a disease, you should talk to your doctor. And I thank you for joining me, because it's been a lot of fun to explore Lifewave and particularly this Eon Patch. So I'm going to, unless somebody has a question about the slides in particular, I'll stop sharing my screen and we can talk. So just let me ask for a minute. Does anybody have a question about the slides? Okay.
Thanks, Nancy. That was great. Everybody can, if you'd like, you can unmute. If you've got some questions there now, you could ask as well. You just gotta go back to the.
I put everybody to sleep. I'm sorry.
Oh, no. I think we're all hanging in there pretty good. Okay, so, all righty. Well, that was really interesting. I feel like I was kind of back in good old science class, and it was, it was very good.
Amazing what they've learned and what they've not learned. I mean, the fact that, they've known about the queen bees, obviously, since before we were children. And they're just getting around to actually trying to figure out what's happening. I'm always amazed. I'm amazed by what they've learned over my lifetime, and I'm amazed by what they haven't learned over my lifetime. So. Yeah.
Well, I heard in ancient times, like honey was just like such a precious commodity. It's just like royal families, were the ones that could really
food of the gods.
Food of the gods, yeah. Exactly.
Having taught history in college, it's actually everybody talks about the Silk Road that went from the east to west, and of course there were incense and foods and whatnot, but one of the foods that was traded along the routes, particularly in areas that didn't have a lot of honeybees, was honey. Well, primarily that was, of course, because it was sweet, and so the aristocrats of various cultures such as China, particularly, as well as Europe, but also parts of North Africa, would trade honey so that they would, because they could sweeten their tea with it, they could sweeten lots of things. Now, of course, it was very expensive, and to this day, it's hard to cultivate. But the wild thing is to know that we've got a pretty sweet patch here. I never thought of it that way.
Yeah,
Good.
a sweet patch. Yeah, that's a
Sweet patch.
good way to put it. Yeah. So, Greg or Chanda, if you're welcome to unmute, and Jill, Eddie, if you have any questions, because we are coming up, I want to respect everybody's time. But this has been great, and Nancy will be back in not next Friday, but the Friday after. And what's the subject on the next one?
Well, I'm not quite sure. Okay, so we did the overview, we did the energy patch, which was the first one, and then we had the 3 YH patches together. So one thing I thought about was, since this Eon patch had so much to do with the wake-sleep cycle, we might do the sleepy nights one to see. I mean, one of the things that I'm trying to sort out, is the differences between the patches, because in particular, they use the same pieces of equipment to evaluate them. And so there gets to be this sense of redundancy for me in the measurements that they take. And you say, okay, well, if this makes you relax and that makes you relax, then what's the difference? And the difference is in the science behind what it is it's targeting.
Right. Well, let's tentatively maybe go with the Silent Nights patch then. That would be good.
Okay.
I think that's a good follow-up. Great.
I know it's hard to know what, there's so many patches. For me, I'm so new to it that it's so, here, let me show.
Yeah, I'm just glad that there's 14 instead of 36.
I know, and what to recommend and that kind of thing. I just have to get a better understanding of all of them.
We haven't done X39 or 49. And I don't know whether I'm saving those for last, but and I'm hoping I'll learn more about X49 before we get to it. But,
is this Aeon one new, or is it one of the
2011?
One of the older ones.
So
that's right, you said that. I saw that. Okay. Okay, cool.
But maybe we can do a little bit of both, maybe, because really the X39 and X49 are kind of the flagship products for the company right now. And so we can just kind of touch on that a little bit, and then, then go into the Silent Nights patch like Nancy was saying. So
yeah, cool.
That would be good. Hey, I want to thank everybody. You guys have been great. I know it's a Friday night, so you guys get your dancing shoes on now. Go do some dancing, or play some nice music.
I appreciate you coming.
Appreciate all you guys. Thanks a lot.
I know. Thanks, you, Nancy. Thank you, John.
Good to see you all.
All right.
Have a good night.
Bye.
Let's see. Where am I?